Hello students, welcome to EPG Pathshala. I am Vidya Kothekar, former professor of biophysics from All India Institute of Medical Sciences, Delhi. I was also professor of bioinformatics at JP Institute of Information Technology, Noida, and professor and director of bioinformatics at Dr. D.Y. Patil Biotechnology and Bioinformatics Institute in Pune. Today, I am going to discuss about a module, double helical structure of DNA and conformational properties of nucleic acids. This comes under the paper Biomolecules and their interactions. Series of experimental observations which preceded the DNA discovery laid the foundation for the discovery by Watson and Crick in the year 1953. This is considered as one of the greatest discoveries of the 20th century. The reason is it opened the gates for understanding relationship between gene sequence and body's vital proteins. Students, let's see what we are going to learn in this module. First, I want to explain you a historical background behind the discovery of DNA structure. You know, no great discoveries in the world are done in a day. Lots of things and many scientific work go behind the discovery. So was the discovery of DNA structure. Next, I want to give you Watson and Crick's model of DNA and its biological implications. I call it model because it was not a structure in the conventional sense where X, Y, Z or Cartesian coordinate details are given. Then we want to discuss the structural features of DNA. Next, I want to explain you general structural features like base hydrogen bonding, grooves of DNA, major groove and minor groove, rotational translational angles in two DNA forms, aDNA and bDNA, sugar pucker and pseudo-rotational torsional angle P, glycosyl bond conformation. Then I want to explain symmetry elements in DNA so that you can yourself generate DNA molecule. Let us see the historical background behind double helical structure of DNA. Many things are there already before Watson and Crick. Like chemical formula was given by Viat and Cohen in 1952. Erwin Chargaff gave, these are called Pareto rules, that any cell organism should have 1:1 ratio of pyrimidines and purines. Studies on sedimentation coefficient, diffusion constant, light scattering, viscosity showed that it is a polymer about 20 angstrom wide and thousands of angstrom long. Its molecular weight was between 5 to 10 to the power of 6 to 10 to the power of 7, and it has approximately 3 into 10 to the power of 4 nucleotides. Chemical structure. DNA molecule consists of a very long chain. The backbone of the chain consists of alternate sugar phosphate groups shown in this picture. Phosphate, sugar, phosphate, sugar, phosphate. Phosphates are joined together by 3-5- phosphodiester linkage. DNA bases These are heterocyclic, nitrogen-containing, aromatic biological compounds: adenine, guanine, uracil, thymine, and cytosine, which we have seen in details in the previous presentation. The structural studies were reported using electron microscopy by Williams in 1952 and Kahler in 1953. These people used freeze-drying method and showed that DNA is 15 to 20 angstrom thick fiber. X-ray diffraction study of sodium salt of DNA had been also reported. These studies were done by taking concentrate of highly viscous gels produced by precipitation of nucleic acids from aqueous buffers through the addition of alcohol by Fuller et al. The rod-like structures formed parallel arrays. These were randomly distributed about the fiber axis. X-ray diffraction studies. Wilkins, Goslin, and Seeds in 1951 and Fraser and Fraser published simultaneously studies on DNA fibers. These fibers showed marked ultraviolet and infrared birefringence, which is characteristics of a long fiber. Franklin and Goslin studied X-ray fiber diffraction pattern of DNA. They found few meridian reflections only. At low humidity, a well-defined X-ray pattern, characteristics of a crystalline pattern, was seen by Wilkins and Wilson in 1953. We show in this picture the fiber X-ray diffraction study of DNA, first by Franklin and Goslin in 1953, which was above 30% hydration, and second on the right hand side by Wilkins group, that was below 30% hydration. In the first photograph, you can see a criss-cross patterns. In the second photograph, you see number of broken arcs, but lot more information than in the first photograph. Let's discuss the Franklin and Goslin fiber X-ray diffraction pattern. A strong meridian reflection at 3.4 angstrom on 10th layer line was seen. Another strong reflection was seen at 34 angstrom. The criss-cross pattern with broken arcs, a typical characteristics of a helical geometry, was seen. Pitch of DNA was equal to 3.4 angstrom and diameter was equal to 20 angstrom. X-ray pattern suggests high concentration of atoms on the peripheral region. Fiber X-ray diffraction by Wilkins group. The interpretation of Wilkins photographs at low hydration was more complicated. It did not have meridian reflections. Its X-ray diffraction pattern had more information content than the Franklin and Goslin's fiber X-ray photographs. Watson and Crick built the DNA picture or a model on the basis of X-ray fiber photograph by Franklin and Goslin, which was taken in 1953, and some features from Wilkins' photograph. They hypothesized that there are two forms of DNA. As any proposed model, DNA must be capable of forming both these structures. Watson Crick Name these two forms as B, which is highly hydrated form of DNA, and A, which is hydrated with less hydration. We show here a beautiful photograph of James Watson and Francis Crick. They are with their DNA model and discussing it. On the right hand side is shown a purely schematic diagram of DNA. The photograph is not as good. The reason is it's taken from the original paper of Watson and Crick. They show two ribbons symbolizing the two phosphate sugar chains and the horizontal rods, the pair of bases showing the chains together. The vertical line marks the fiber axis. The two chains of DNA molecule, according to the Watson-Crick structure, are coiled around a common axis and joined together by hydrogen bonds between the bases. Both the chains make right-handed helix. The sequence of atoms in phosphate sugar backbone runs in opposite direction or anti-parallel. Phosphate and sugar are on the outer side. The bases are inside. The distance of the phosphate from the helical axis is about 10 angstrom. What is plectonomic and paranemic winding? Watson-Crick described both these windings. Now, these are the two ways by which helices wind over each other. In a plectonomic winding, the one described earlier, and in the paranemic winding, when the two coils are put side by side. Watson and Crick preferred to describe DNA using plectonomic winding. This was in spite of great difficulties with plectonomic winding. Why? Because this gave successive base pairs in equivalent orientation, and it was very difficult to be tampered because paranemic winding can very easily separate and the DNA code will be destroyed. A genetic code should not be destroyed. So, Watson and Crick preferred plectonomic winding. Base hydrogen bonding. Watson and Crick examined number of edge bonding schemes between A, T, G and C using cardboard models. Two schemes with A pairing with T and G pairing with C with 3 hydrogen bonds gave suitable orientation of the glycosyl bond. You can notice the glycosyl bonding position R in AT base pair are almost similar to that in the GC base pair. Major and minor grooves. Orientation of the glycosyl bonds in the Watson-Crick model is cisplanar. This divides the area around the helix axis asymmetrically, giving rise to minor and major grooves. We show in this picture the minor groove and the major groove. In the minor groove, the 5' strand is lower to the 3' strand. Base complementarity. The base complementarity was inbuilt in Watson and Crick's model. According to it, A can be paired with T and G can be paired only with C. This was in accordance to the Chargaff's rule. Latter suggested that in any DNA model, the percentage of A is almost same as percentage of T, and percentage of G is close to percentage of C. The greatest outcome of Watson and Crick structure is its biological implication of their model. According to them, a genetic material, its main property should be that it should replicate. Second, it should have highly specific influence on a cell. Watson and Crick did not much clarify the second. However, they explained the basic principle of replication and the fidelity of the copying process is because of the necessity of precise orientation of the glycosyl bond that leads to only a specific base pair formation, because of which A can only pair with T and G with C, T with A and C with G. So if you have AATT, you will get TTAA and a carbon copy will be made. DNA structure at atomic resolution. Wilkins group developed a linked atom least square refinement method for fiber X-ray diffraction pattern. The atomic level of DNA structure were given by Arnott et al. In 1976. The gross structural features in Watson and Crick's models were preserved in both these structures. Glycosyl bond orientation and H bonding between base pairs. We show in this picture hydrogen bonded base pairs AT and GC. We can notice the orientation of the glycosyl bonds NR, NR in both the cases. The distance between C1' C1' atoms in the opposite strand was equal to 10.85 angstrom in both the cases of AT and GC base pairs. The angle between the C1' C1' and glycosyl bond was 51.5 degrees, which leaves the bases in identical orientation with respect to DNA backbone. In the case of AT base pair, the hydrogen bond was between N6H of adenine with O4 of thymine and N1 of adenine with N3H of thymine, whereas in case of GC base pair, O6 of guanine hydrogen bonds with N4H of cytosine, N1H of guanine hydrogen bonds with N3 of cytosine, and N2H of guanine has H bonding with O2 of cytosine. The backbone geometry. The distance between consecutive base pairs, usually called as base pair rise or TR, is 3.4 angstrom in bDNA. The pitch, the distance among the helical axis of the DNA bases with identical orientation, is equal to 34 angstrom. The bases are in plane and perpendicular to the helical axis. The backbone geometry. Each pair is rotated by approximately 36 degrees about the helical axis with respect to the preceding base. This is generally called as a twist angle or a helical twist. bDNA has an overall width of 20 angstrom. aDNA was paracrystalline in nature. It had average width about 22 angstrom and 11 base pairs per turn. The base pair rise PREZ was 2.56 angstrom and the twist angle was 32.7 degrees. Nomenclature for backbone atoms. Backbone angles are defined by taking a sequence of 4 consecutive atoms from the backbones O3 P, O5 C5 C4 and C3 Furano's angles are defined by taking 4 atom sequence from 5 sugar atoms C1 C2 C3 C4 and O4 Backbone angles are given. PO5 is alpha, rotation around O5 C5 is beta, rotation around C5 C4 is gamma, C4 C3 is delta, C3 O3 is epsilon, and O3 P as zeta. Whereas sugar angles are given as gamma 1, gamma 2, gamma 3, gamma 4, and gamma 0. These are described in the subsequent slides. Torsional or dihedral angle, as discussed earlier, we consider a 4 atom sequence J, J dash, J2 J3 and clockwise rotation of the bond J, J dash or a near bond about the bond J dash, J2 dash, that is the central bond, to bring it in the direction of J2 dash, J3 dash, that is a distance bond. We take it as a positive dihedral or a torsional angle. We define in this the backbone, sugar and glycosidic torsional angles for B and A form of DNA. We have also given their values for B form and A form of DNA, and you can notice the similarities as well as differences in these values. For example, angle alpha is always around minus 40, minus 50, or we call it minus G conformation, whereas angle zeta is also high minus G conformation, we can say minus 90s and so on. Beta is somewhere minus 150, gamma is always plus G, that is 30 plus and minus 30 degrees rather. Delta angle doesn't change, but it depends on the sugar conformation. It is minus 156 degrees in BDNA and 83.5 degrees in ADNA. The reason being, in the case of BDNA, we have C2 endo pucker, and in ADNA, we have C3 endo sugar pucker rather. Sugar pucker nomenclature. As we have discussed earlier, we imagine a plane through C4 O4 and C1 atoms, the C5 atom of the backbone, and N, that is N1 of pyrimidine or N9 of purine, lie on the same side. Atoms C2 and C3 can lie either on the same side or opposite side, giving envelope and twist conformations. In the twist conformation, one of the atoms may be pushed more out of plane. If C2 is pushed more, we designate the conformation of the sugar as C2 endo. If C2 is pushed more in opposite direction, then we name it as C2 exopucker. Similarly, one can have C3 endo or C3 exo sugar pucker conformation. Sundarlingam's group, Altona and Sundarlingam, they devised a very interesting way by which all different sugar puckers can be linked through a pseudo-rotational angle, which is called pseudo-rotational wheel, and one can describe all conformation like C3 endo, C3 exo, C2 endo, C2 exo, and so on this wheel structure. And from the value of the pseudo-rotational angle P, one can calculate all 5 angles in the sugar ring also. Pseudo-rotational angle. As we saw, the sugar puckers go as C2 dash and O, C3 dash and O, and like that, not any continuous angle. But it was the great idea by Sundarlingam's group. Altona and Sundarlingam showed that there can be a pseudo-rotational angle, P, and which can be calculated from the 5 cyclic sugar torsional angles. And they gave a relationship, mathematical relationship, by which this angle can be calculated. And also they gave another relationship by which, if you know the pseudo-rotational angle, you can calculate the 5 torsional angles and use them. The symmetry elements in B and A DNA. We show in the picture the view of a one base pair of DNA in the XZ plane. You can notice that sugar phosphate backbone of the root strands grow in opposite direction with respect to the Z axis. We show another view of the base pair that is in the XY plane. We show helix axis by a dot. In this case, x-axis is a dyad or a pseudo-dyad axis. Dyad because the backbone is having a mirror symmetry around that axis, and it is called pseudo-dyad. The reason is the bases, because these are not identical, they are not mirror images. But if you imagine the same base on the opposite strand, then it will be mirror image. That's why we call it a pseudo-dyad axis. Generation of DNA using cylindrical polar coordinates. Like, there are various ways by which we can generate DNA molecule. One can use Cartesian coordinate building blocks, or more conventional is to use cylindrical polar coordinate building blocks, where the cylindrical polar coordinates Ri, theta I and Zi for each atom in the block are given. Now, using the value of twist Tw and translation along helix axis Tr, one can calculate the cylindrical polar coordinates of any residues in the same strand using the first equation, or any residue in the opposite strand using the next equation. The Cartesian coordinates Z are of course calculated by the first two equations, but x and y can be calculated simply by using xin equal to Ri cos of theta in, and yin is Ri sin of theta in, or theta in dash if it is a opposite strand, and one can generate any DNA polymer. So students, what did we learn in today's module? First, we saw historic developments prior to the discovery of double helical structure by Watson and Crick. Then we tried to understand the basic Watson-Crick's proposal, its biological implications, and existence of polymorphic forms of DNA, B and A form. Next, we tried to understand the details of structures of B form of DNA, which is a highly hydrated form, and A type form of DNA based on the fiber diffraction data, single crystal X-ray diffraction on oligonucleotides and milli-helices, and linked atom least square refinement procedures. Later, we tried to understand IUPAC, IUB nomenclature for rotational, torsional angles in the backbone of the DNA, and the bases and sugars. We studied base hydrogen bonding, groove dimensions, backbone torsional angles, sugar pucker, symmetry elements in double helical structure. Lastly, for those who are interested in modeling DNA by themselves, we have given a mathematical expression needed for the simulation of DNA. So you can write your own software and make DNA from any starting data, either cylindrical polar coordinates or Cartesian coordinates. And I thank you all for kind attention.